Thank you so much, Sarah, and thank you all for having me here. This has been a really outstanding education for me, getting up to date with some things. I'm going to reintroduce myself. I'm a relic from the past. I was a caveman on the paleo diet, if you will. In any case, I found out early in my career I was challenged some things that turned out to be quite a challenge. Basically, I was challenged our reverence for protein, as you'll see in a moment, and I was also using experimental animals. So I'm going to discuss with you some evidence from the experimental animal literature. And I know that's not exactly kosher, if you will, but nonetheless, I spent a lot of years adequately funded by NIH and through those years, and got into exploring a question that actually illustrated for me some things about the basic science of nutrition. Let me tell you what it really was all about. I started out early on, after being raised on a dairy farm, by the way, and doing my doctoral dissertation on trying to figure out how to grow animal protein more effectively. I got involved in a project feeding malnourished children in the Philippines, where we wanted to actually promote the consumption of more animal protein. It was in the Philippines, but then I happened to see something that was really odd, not believable, but basically I was told by my medical colleagues that they were operating on some children with primary liver cancer. I was totally off the chart. I didn't really believe it. In fact, I learned it on the golf course, so I didn't take it too seriously. But in any case, I remembered that quite well when this paper here came out. This is a paper that was done on experimental animals from India, in which they were actually wanting to have a look at what was causing primary liver cancer, and they were using a rat model to do that. And so what their hypothesis was that the high protein diet would actually tend to attenuate and slow down the development of that cancer. That was their idea. That would have been the idea of all of us, basically, that protein somehow would have been protective. But what they actually found, and they didn't quite believe it, went on about their work later on, but in any case, it turned out that with a modest increase in protein in the diet, from 5% of calories to 20%, those animals, after being exposed to a chemical carcinogen, just like the 5%, those animals are the ones that got the cancer. It's really quite dramatic. We didn't need a statistician in those days, I think, to determine whether that was interesting or not. So then that was the question that got me into this field early on. It was this idea that just not quite believable. But in any case, I wanted to get into this based on what I thought I was hearing about the children from my friends, together with the evidence here coming in these experimental animal studies. I basically wanted to do two things as far as the research was concerned. First off, I wanted to confirm whether it's really true. Did this work from India, was it reliable? And secondly, I wanted to learn something about the mechanism by which it worked, if it were true. And so what it got me into is really the area I like most, that's basic science and the biochemistry of things. And as I looked for the mechanism with my students over the years, I really found myself involved in that concept of determining biological plausibility. So much of my career was actually focused on that idea, determining whether a relationship, or really an odd relationship like that, was biologically plausible. And I did that as we went through the years looking for essentially principles that turned out to have wide application, not just for protein, but for other nutrients, other cancers, other diseases. And so let me show you what I mean by, as we walk through, learning some principles, which I think are still kind of fascinating even today. So here's one display of some of the evidence. Here's a rat study. Animals exposed to a chemical carcinogen to initiate the cancer, if you will, the mutagenesis. Then subsequently feeding different levels of protein. Looking at the formation of early cancers, we call them pre-neoplastic foci. 20% protein diets, of course, you can see a generous increase in the growth of these early cancer cells. 5% nothing. Again, the same thing that the Indian workers had done, so I thought, well, we're getting on the right track here at least. Then there was, in a number of studies after that, we got into looking at how significant is this protein effect on that particular cancer lesion. So we did a back and forth kind of study. First 3 weeks, we fed 20% protein, 5%, 20%, and back to 5%. And we did this incident with fat and pancreatic cancer and some other things, and it led to a concept that I found very exciting, that we could turn on and turn off experimental cancer, even though it's experimental animals. We could turn it off, turn it on, and turn it off by nutritional means. So that's one principle that I have kept through the years, that nutrition is a significant factor in controlling the development of cancer, at least experimentally in laboratory animal systems. But then we followed it up. That was early cancer, so I wanted to see what eventually would happen if we did a whole lifetime study. This is the lifetime study. Same 5%, 20%. Both sets of animals exposed to a carcinogen to give rise to liver cancer. Fairly good size study for this kind of study. And you can see at the end of the 2 years, 100 weeks, the animals given the 20%, they had a generous amount of tumors compared to the 5%. At the end of 2 years, what's the normal lifetime of the animals? The 5% animals all living, thrifty hair coats, energetic, as if they were still reasonably young. In contrast, the animals given 20% were all dead with liver cancer. Again, so short-term observations, just early cancer, long-term observation for the whole lifetime, just changing the protein level within, one could argue, within reasonable limits. That's the difference they saw. So then it was that I was just in those days just thinking about protein, but was actually using the protein that was available to us in experimental research, and that was casein. I was raised on a dairy farm. So all of a sudden come to the realization, hey, we're using casein here to turn on that cancer. That took me a couple of years to digest that, no pun intended. But in any case, so we decided to try soy and wheat protein. Even when soy and wheat protein were fed it up to the 20% level, no response. So all of a sudden it was emerging here, this distinction between animal and plant proteins, if you will. This is something we already knew from other kinds of outcomes and functions, that animal protein does function differently than plant proteins. Then we got into this question that I found really exciting. As I said, I wanted to know the mechanism by which this worked. Now here's a timeline of the way we sort of consider experimental carcinogenesis, going from initiation, first stage, promotion, on into progression, initiation being the stage during which the mutation is occurring. So we spent about 15 years actually looking for the key rate-limiting mechanism. I mean, everybody wants to know what the mechanism is, if you will, in those days. And it turned out every time we looked for a mechanism, we found one when the protein level in the diet was higher. So high protein diets inhibited the entry of the carcinogen into the cells. It increased the activation of the carcinogen by an enzyme system that's very complex and did a lot of things to the enzyme that I found really fascinating. It increased the binding of the DNA, which leads to the mutation if it's not repaired. Actually, the high protein diet actually compromised the ability of the cell to repair its own damage. So then we looked into the promotion stage, still not putting our finger on the key rate-limiting mechanism. And what we learned was that the high-protein diet stimulated cell replication. Later learned it was associated with the production or synthesis of hormone, the insulin-like growth factor. And again, the high-protein diet decreased the ability, the normal ability of the body to repair the damage. That is through the production of natural killer cell activity and a bunch of other things. So I finally got to the point, this is frustrating. Where is this mechanism? Finally realized there is no such thing. And it opened up a whole new world of thinking for me as far as nutrition is concerned. When a nutrient is consumed, there's a whole tsunami, I would say, of countless mechanisms operating in synergy, which is a really fascinating concept. And then you put them all together, you get the effect that it's much more comprehensive than what we generally think in a reductionist way. Now, here's another thing. Incidentally, this occurred, as I say, 35, 40 years ago, before the Human Genome Project, and we had our own way of sort of looking at the gene activity. But here's a little scheme, if you will. Taking normal cells, this is just a cartoon, if you will, creating on the left-hand side some cancer genes through mutations. On the right-hand side, producing a lot less. So you got high genes on the left, a lot of mutated genes on the left, not on the right. Under that circumstance, of course, you say, oh, okay, genes, if you will, damaged by the carcinogen is really what determines the outcome. However, we can say genes cause cancer. We know it starts with a genetic mutation, but in reality, the level of protein was far, far more significant than the genetic dose, if you will, which opened up, again, a principle for me. Nutrition controls genes irrespective of immunogenic dose. And that's what was an extrapolatable idea, in my view, for a lot of disease conditions. One more, I wanted to know, I just said the 5%, 20%. We did a couple of other things along the way, but I really wanted to get a little better fix for the level of protein that might actually turn on the cancer. So we did, as you can see here, different groups, 4, 6, 8, and 10, 12, 14, and up to 20, you can see. And so what turned out was really interesting, especially the hockey stick effect that we can see here that's typical of nutrient activities, by the way, up to a certain level. Even though the animals had been exposed to a heavy dose of a carcinogen, up to a certain level, no tumor development. Only when the amount is exceeded that the animals normally need. That's when the cancer starts to grow. And that's also the same time when some other things go too are not so good. We humans are in that red zone. I see a red zone here on my thing here, so I'm going to have to quit. But in any case, dietary protein in excess of the recommended dietary allowance, which incidentally in the case of humans is around 10% of energy or so. That's when mischief starts. And I wish I had the time to tell you, but it's a whole world. It's a very different world of thinking about what nutrition is and how it works. Not just in this very narrow little study involving liver cancer in rats, if you will, but in a much larger context. And when I got done after all these years looking at a whole series of mutations, I had no preference for assuming that, I was working toward a position about diet that was based on ethical considerations. I was just wanting to do the science. That's all. The entire operation was just based on the science, having a fascinating time of learning to see where these questions took us, and that was the reason I came to the conclusion I did. And along the way, in the interest of time, I'll have to just make one comment here. I think that one of the corrupting influences in our thinking over the years as far as nutrition and disease outcome is concerned, one of the compromising things that we tend not to think about is what effect does protein have. And when I speak of protein, I mean that plants have all the protein we need. When we start getting more protein just because of our reverence for that nutrient, usually by consuming animal-based foods, that's when the problem starts. And the higher protein consumption, quite frankly, a dose-response relationship when you go above the amount needed, the higher protein consumption actually has direct effects on the protein, as well as equally significant, if not more so, indirect effects by the fact that the higher protein consumption under a zero-sum game with calorie intake, the higher the protein consumption, the lower the consumption of the foods that matter. So it's a combination of direct effects causing damage and the absence of indirect effects, its indirect effects by simply deflating the consumption of those things that really matter. Thank you.